Good morning, everybody. Welcome to the last day of this summer school. Our first speaker is Carmen Pin. Dr. Pin is a senior director in the systems medicine team of the clinical pharmacology safety science at AstraZeneca in Cambridge, United Kingdom. And prior to joining to AstraZeneca in 2018, she led the computational gastrointestinal biology group at the Quadram Institute Bioscience in Norway. She studied veterinary medicine and also did her PhD in veterinary science, and later took full undergraduate degrees in statistics and mathematical statistics. Carmen has co-authored numerous scientific articles and led some substantial scientific research projects funded by the European Union and the BBSRC of the United Kingdom. So welcome, Carmen. The floor is yours.
So I would like to, I'm not sure if we can interact, but well, I will try to if you have any question, or I'm not sure if you could ask for a microphone and interrupt me, but that would be, I mean, if that's possible, that would be great during the talk that we chat a little bit. I mean, that is not only me just telling you for an hour what we are doing, but that I can have your feedback. So why do we need models in biology? Well, biology is very dynamic. Even when it seems it's not changing, everything is changing, but it's in a way that everything is stable. So what this means is that when we approach a biological system, we have many moving parts interacting with each other. And this is something very challenging for us to put together all these moving parts and how they interact with each other in our mind is really difficult. So for this reason, to do this, to achieve this integration of dynamic parts, we have mathematical models. And so these mathematical models can summarize all these dynamics and different parts moving together and interacting with each other and help us understanding what are the outputs, what are the inputs, and how the system is working. So we can have very different behaviors in these dynamic systems. And for instance, in the case of, let me see if I can use the laser point. So in the case of populations like the polar bears, well, we know that if nothing changes, the populations are more or less stable. And this can be modeled with these simple logistic models in which you have a population that increases and then it reaches a number in which is in balance with the medium, so with the carrying capacity of the medium. So and once it's there, the population, the number of the population varies a little bit from one year to the other, but it remains stable. A very different situation is, for instance, these lynx and hare populations. No? In this case, we have a prey-predator system in which the lynx eats hares. So when the lynx increases, the lynx is the red one, this is data, the hares are going to decrease because they are eaten by the lynx. But when the hares decreases, the lynx also has to decrease because it is not going to find food. So we have these two systems oscillating with each other. I mean, these two quantities, the prey and the predator numbers, they depend on each other and they are oscillating in the environment. They are very different, these dynamic systems. No, one is a steady state, the other one is an oscillatory system, but both can reach equilibrium. That means that they will be, in this case, they are stable. The equilibrium is stable. There are other systems in which the equilibrium is unstable, but in this case, it's stable. So So we have systems with many different behaviors, and one of the most interesting behaviors is this chaotic behavior, this chaos theory that started with Edward Lorenz when he was modeling the climate. He was forecasting the weather, and then just by chance, he discovered that very small changes in the code, rounding errors in the computer were causing very big changes in the output of the predictions. So what he discovered was that, you have a system and very, very small input changes, so that should practically drive very, very small output change, change completely the behavior of the system, not only the quantity or the prediction level, but the complete behavior, what is from being a steady state to be an oscillation or so. So I will show you how this works. And that was extended, this chaotic behavior was later found also in many biological systems. And one of them is this population, this biological population that are the cicads. Now, the cicads is this animal. And one peculiarity on this population is that the generations don't overlap. That means that the cicads bury the eggs of the next generation, and those eggs emerge only in 13 or 17 years. So this type of population then can be modeled with what is called a logistic difference equation that is similar to the logistic differential equation. Sorry, I think my camera has stopped working. I want to see if I can restart it now. So these cicads the idea is that you can model these cicads with a logistic difference equation. It's a discrete equation. And in that equation, if you have a slow growth, you can see that the behavior is as we describe, first an increase until you reach the carrying capacity of the environment, and then a steady state. If you increase the proliferation of that, or you decrease this emergency time, the proliferation rate of the population, what you get are a population with stable behavior, but cyclic behavior. It's you have constant oscillations that are repeated over time, and as you increase more the growth, the proliferation rate of the population, these oscillations will remain stable, but they have more than two stable points. But there is a point in which you keep increasing this proliferation rate, and what you see is an unpredictable pattern of oscillations or changes in the population. And that's what is called chaotic behavior. That means that, you get here a peak, then you get another peak, then it goes down. But you can see there's no pattern in these changes. And that's what is called chaos. So the same system, the same simple deterministic model, depending on the value of one parameter, it can have many different structures, or can describe many different behaviors. So after this discovery by Lorenz and May, chaotic fluctuations have been detected in many other biological systems, like the heartbeat intervals, the cardiac system, or also the neuron firing intervals in the cortex. So it's quite a common dynamics in biology. So also a mathematical model help reveal how the neuronal electrical impulses are transmitted in the cortex. So, and this was the work of Hodgkin and Huxley. I don't know if you know, Huxley was a very important scientist in the 30s, 40s. He got the Nobel Prize for his work together with Hodgkin, but also Huxley, which is very interesting because he was the brother of Aldous Huxley, and Aldous, his brother, wrote this book that was very famous at that time, that is called Brave New World. And well, it's a futuristic science fiction book describing a future society. So it's quite interesting to read it because it describes how the society would be in the 90s. The book was written in the 30s, I think. So and it's quite interesting to see that he got right many things. You know, it's well, it's in the hindsight, it's a nice book to read. But well, that's not our presentation today is about something different, and it's about his brother Huxley, who together with Hodgkin, what they did was measuring the membrane potential in the giant squid axon, in this big animal. And so what they did was using mathematical models, using equations, they could describe a hypothesis, and it was a hypothesis comprising 4 independent ionic channels. And with that, they were able to describe the data they were recording in this in the membrane of the giant squid axon. So this was quite interesting because they got the Nobel Prize, but also they started a new theory in ionic dynamics. So ion channels have been studied very much in detail since then. For their study, you really need modeling because this is one of those systems that you cannot really handle only with data in your mind or with simple operations. You need differential equations. And well, this work is the base of the current ionic studies, of the current studies of the current theory in ionic dynamics and also in electrophysiology, both in the heart and in the nervous system. Another important discovery that it was based on mathematical modeling, for which mathematical modeling was really important, is the circadian rhythm. So we know that there's a circadian rhythm. We understand that because we know that we go to sleep at night and we are awake during the day. And many of our physiological activities are also regulated in this way. They change from day to night. This group of people, Hall, Rosbach, and Young, help to understand is that this circadian rhythm can be coupled with the light, but it's independent of the light. I mean, you have these cycles of behavior of all physiological activity, even if you are in a complete darkness condition. And that's because you have group of proteins oscillating in a timely manner, in a very organized, timely manner. And then they discovered this group of proteins and that they were mainly regulated, or these oscillations, these stable oscillations over time, were mainly regulated by this translation feedback loop, in which, well, they name 2 main proteins called period and timeless. And when they are translated and transcribed, when they are synthesized, they form a complex that inhibit their own translation. And that this feedback loop is essential for the oscillatory behavior of this system. So it's very complex system, and there's a big variety of models to describe it, from very simple to much more complex, depending on how many elements they consider. But well, the mathematical modeling mathematical models are really essential to understand and handle this kind of system. So now I would like to, so we are talking about modeling, and I am talking about, well, equation and systems that are complex. So what is this type of modeling about? Well, it's about understanding the law of the systems and describing those laws with a computational algorithm or with a set of differential equations. But the basic is the basis of this activity, of this modeling activity, is understanding the laws, is to develop these models. So a little bit, I brought here this video that is part of a lecture in physics by Richard Feynman. I don't know if you know him. This also a Nobel Prize in Physics, very, very clever man. And well, there are many books. There are many serious papers about his work, but also kind of popular books about his tricks. Because he was a great personality, not only professionally, but also a very good lecturer and quite a funny man. And then I would like to share with you this video, see if I can play it.
In general, we look for a new law by the following process. First, we guess it. Then we well, don't laugh, that's really true. Then we compute the consequences of the guess to see if this is right, if this law that we guessed is right, we see what it would imply. And then we compare those computation results to nature. Or we say compare to experiment or experience. Compare it directly with observation to see if it works. If it disagrees with experiment, it's wrong. In that simple statement is the key to science. It doesn't make a difference how beautiful your guess is. It doesn't make a difference how smart you are who made the guess or what his name is. If it disagrees with experiment, it's wrong. That's all there is to it.
So I don't know, just in case if you didn't, you couldn't listen very well. What he says is that the way to go on about this model development is, or this understanding this type of laws governing these dynamic systems is that first you guess what's happening, what's the main law driving the process, and then you compute it, and then you get some predictions, and you compare predictions with observations, and if they don't agree, the model is wrong. And that's and although that when he's saying that, or when I say that, you may think, well, it's so common sense, and why do we need to listen to this? The truth is that when you are working, that's a very, very common situation in which the modeler doesn't want to admit that his model is wrong, and they blame the data or the experimental error, or that they need more data, or that these are kind of funny situations when you are working. So let me now, I don't know if I will be able to get some kind of input from you, but this is a small exercise about just to show you why do we need quantitative models in biology. One reason is what we have been speaking, what we have been saying. Some processes are extremely complex, so you cannot really put in your mind all those moving parts together and the interaction between them and get an output without a computer, because your mind is not able yet to do those things. But another reason is that even for simple processes, quantitative models can be extremely helpful. And so let me show you an experiment. This is an experiment that Amos Tversky and Daniel Cunningham did. It was an experiment, a social experiment, using university students. What they did was asking the students what is a more probable event. The Linda is a person, is a bank teller, or that Linda is a bank teller and is active in the feminist movement. So now I would like you to vote and in a way say, I don't know if I can get any feedback from you. At this moment, I cannot see you. Just to, for instance, hands up who think that the more probable event is that Linda is a bank teller. So I cannot see you, so I don't know. And hands up, the one that the people that think that Linda is a bank teller and is active in the feminist movement. This case 2 is more probable. So well, I cannot see you, but then let's go to why I'm talking about this very simple thing.
There were 10 hands for number 1, no? For number 2? For number 2, nobody.
Nobody, okay. So you are, that's very good. That means that this course was excellent because, yes, that's the right answer, of course. I mean, and that means that you are probably quantitative people. I mean, you have quantitative minds, because it's true that Linda is a bank teller is more probable because this set will be included in the set of events that I mean, it's much more probable that Linda is only a bank teller. That if you make we put another condition, then we are restricting the probability. No? So of course, it's more probable. However, when Amos and Daniel made this experiment among university students, what they found was that most of university students thought that the option 2 was more probable, and they thought that because this was more appealing to them. Because their perception was, well, Linda is a bank teller and active in the feminist movement. It's like Linda is more real, is more of a real person. So the thing is that our perception is biased. Oh, sorry, our perception is biased, and one of the things that is not biased is a mathematical model. I mean, if you can quantify the events, you can see that this set of events is much bigger than that one, than the second set of events. So because this set of events is much smaller, and in fact inside the set of events, the number 1, it will be much less probable. So if you can, that's in a probabilistic way, but there are many other ways to quantify what is more probable. And if you quantify, you will always get the right answer. If you don't, and you just let your perception tell you, or what we say, your intuitive thinking tell you what is more probable, you may be wrong. I heard something in the. Is there any question or any? Okay, we keep going. Okay. So now let me talk about modeling pharmacology, because this has been a little bit of an introduction about models in general. So in pharmacology, the first thing is that we use a lot empirical models. So far, we have been talking about mechanistic models, models that are describing the process, the mechanism, or the main laws driving the system. So empirical models are just based on data. They describe data, they interpolate in the data. So if you don't have data, you don't have the model. Mechanistic model, as you have been seeing until now, or what I have described, is trying to understand what is the nuts and bolts behind the process, and implement that in a computational or mathematical way to get predictions. So in the past, not so long ago, but in the past, modelers, all of us, we were very proud to do mechanistic models, and we, in a way, we were dismissive about empirical models, because practically it's data interpolation. It's the empirical functions with the shape of the data, or interpolating between data and data, between data points. So, however, currently, with the introduction of artificial intelligence and the new technologies that generate large data sets, we are not dismissive anymore, because this has become a very, very important and essential scientific field of research field, that this empirical modeling that now is within artificial intelligence and machine learning is really important, and it has a huge impact to understand large data sets, so what information is in there. So still mechanistic models, of course, we have better prediction performance, and that means that we can extrapolate. We don't need the data. If we have a good model, we can give predictions for situations in which that we cannot measure, and also we can investigate how the process works. We can test hypotheses, what hypotheses match the data. However, the new AI and machine learning algorithms are also very important to discover hypotheses and information that is included in these very large data sets that we cannot navigate. Now we have new technologies like omics, transcriptomics, proteomics. You get thousands of proteins and you practically are flooded by the system. I mean, it's impossible to understand what to visualize or just get the information in those data sets without this type of technology, without AI or machine learning. So we are not dismissive anymore about empirical modeling or AI. In fact, is quite important. So, but in the pharmaceutical industry, the traditional empirical models, just based on the data, quite easy to produce, or well, straightforward to produce. Once you have the data is still very much used. So let me show you first here in this part what is the drug development pipeline. So what they do is, what we do in the companies is just select the target, not those that molecule that we want to modify. For instance, if it is cancer, maybe we want to inhibit a protein of the cell cycle, or maybe we want to disrupt DNA synthesis during replication, so that we disrupt proliferation and we disrupt cancer cells of dividing. Then, well, you generate a series of compounds with that activity because there are these chemical, there are several probably chemical possibilities to do that. And then you just optimize that chemistry and go to preclinical phases in which you use in vitro and in vivo systems to assess the safety and efficacy of those compounds. And then after that is assessed and it's all clean and good. Green is because well, it doesn't have any issue. Then you can go in clinical trials with real patients. So and during this pipeline, we use modeling a lot. So one of the things maybe when you finish this course, you should have in mind is that the pharmaceutical industry can really offer interesting jobs in modeling and in numerical approaches and good jobs. I mean, it's a good it's a good field to be in if you have a numerical interest. So this, well, but talking about again empirical models and AI. So, for instance, in target selection, AI algorithms are now very common to identify new compounds, or to design compounds that can affect a target, a molecular target. So then during the lead optimization and the preclinical assessment of drug safety and drug efficacy, there's a lot of empirical modeling, just connecting the exposure with the response. No, for instance, if you have a data in a preclinical species, in rats or in monkeys, so you can connect the dose with the response that is either a toxicity response or an efficacy response. And also during the clinical phases, once you have the patient's data, it's also very common to get the data and to connect exposure and concentration of drug in plasma with the response. That means maybe the tumor shrinking, how the tumor is, if it is oncotherapeutics, how the tumor is reducing, or with toxicity, with adverse events associated also with the drug. That in the case of cancer treatments, the adverse events are quite remarkable. So it may be decrease of platelets, or it may be gastrointestinal events. So, and you can you can understand in a quantitative manner how the drug is producing this efficacy or toxicity endpoints. So when we talk about empirical modeling in pharma, many times we say PK/PD approaches. By that, we mean pharmacokinetics-pharmacodynamics approaches. And just to a first explanation about what we mean with that. Well, pharmacokinetics relates, is the process in which you relate the dose that you give to the patient with the drug tissue concentration. So it includes all these events of absorption, distribution, metabolism, and excretion of the drug. In a way, in a very simple way, pharmacokinetics is just embraces all those events or the processes that well, the processes in which what the body does to the drug. No, that's the processes in which the body is doing something to the drug, and what it's doing is either absorbing, distributing, metabolizing, or excreting the drug. So, and this is what we call pharmacokinetics. Pharmacodynamics is what the drug does to the body. So once the drug is in the tissues, it's going to disturb. I mean, if it is an adverse effect, it will disturb either the liver functionality or the GI functionality of the lung. If it is an efficacy activity of the drug, it will disturb the tumor growth, or maybe it will correct a disease process, not something that is not working very well in the liver or in the kidney will be corrected by this drug. So we have a PK, what the body does to the drug, so that the drug comes into the body and starts being metabolized and cleared from the body. And PD, that is what the drug does to the body. I mean, how the drug that is in the tissues is going to do in all these different systems and organs that we have. So also very important in the in pharmacology is to understand what is the effective concentration to achieve what we want with that drug, and what is the toxic concentration, that concentration at which we start seeing toxicity. So there's no, I don't know if you know the expression of everything depends on the dose. So depending on the dose, so if we give some dose to a patient, you are going to have a concentration of that drug in plasma. So depending on that dose, at this dose that we have in the blue line, what you can see is that we are in a very good situation because we are above the effective concentration. So we are going to have the effect that we are targeting with this drug, but we are not reaching the toxicity concentration. So we are in a safe region. And this is what is called the therapeutic margin. That is, you are above, you achieve efficacy, but you don't reach toxicity. So, and this the basis of this is very important in pharmacology maybe before you move to clinical trials, and even after you move to clinical trials, you need always to assess this. However, when we do this type of assessment using PK/PD or empirical models that connect, that need the data, well, it's not an optimal situation because first, we have a very limited predictivity because we need the clinical data. So if you need the data to predict the data, your predictive ability is not great. And also the first clinical trials in which you don't have data are quite dangerous in a way because you will base these clinical trials on in vivo and in vitro data, but with this type of models that are empirical, just data, they describe the data, you may have failures because there are cross-species differences, and the models, because they just follow the data, will not account for those cross-species differences. So, and there have been very big failures in clinical trials because of this type of approaches. So then to assess this, the safety and efficacy of drugs, that is this plot that we were looking at in the previous slide, we need to improve this type of modeling and to move from these empirical models to more mechanistic models that offer predictions prior to the clinical trials. So this would be the PK part, that part of which you give the drug to the body and the body tries to get rid of the drug, that is called the PK part. And here empirical, we have what they call empirical models are called PK models, and more mechanistic are called PBPK, that stands for physiological based pharmacokinetics. In what is called the PD part, the pharmacodynamics part, in which we have what the drug is doing to the body, know how to describe that. The empirical is called PD models, and the more mechanistic is now called quantitative systems pharmacology or quantitative systems toxicology models, if they describe either efficacy or toxicity. So we are trying to move from empirical to the mechanistic in both parts, both in the PK and in the PD, and in the distribution of the drug in the body and in the response of the body to the drug. This is an example of how the comparison between PK versus PK models. I mean, the PK are the empirical models that just connect data with connect the data, and the PK are models more mechanistic with a series of hypotheses and then with more predictive, with better predictive performance. But in both models are describing the tissue concentration dynamics after dosing a drug. So it's this that what the body is doing with the drug. So in the empirical model, usually we have just two compartments. One is central, that represents usually the plasma, and the other one peripheral. Sometimes even we have only one compartment. So they are very, very simple, and they are handled as ordinary differential equations, and they are based on data. So you really need the clinical data to establish this type of model. For the PBPK model, well, it's a model in which you consider the administration of the drug, how the drug distributes through the different tissues, and is metabolized by some of these organs. So they are complex models with multiple compartments, usually described by a large system of differential equations, and then you need a computer to move all these different systems at the same time. But so it's more complex, but on the other hand, because it's based just in the patient's characteristics and in the drug properties, you can predict the concentration of the drug in the body before the clinical study starts, I mean in advance. So, and that means that it's quite much more powerful, more difficult, more challenging, but more powerful. I mean, you have the chance to give a proper prediction. So we are in the same case for the pharmacodynamics part, not this PD versus QSP, QST models, the organic response to the drug. No, that's what we are measuring. What the organs, what the drug does to the organs. So when we have empirical models, usually they just connect the symptoms with the concentration in plasma, or sometimes even just with the dose. And you have closed forms, differential equations, probabilistic models. I mean, but usually they are quite simple. When we move to quantitative systems models, what we try to understand or to describe with our models is the pathophysiology and the drug mechanisms of action. I mean, how the drug enters in the cell, disturbs at the molecular level the DNA synthesis or a protein, and how that disturbance at the molecular level propagates to the cell, to the tissue, and to the body to give a symptom. And we need to understand the dynamics at all these different scales. So they are much more complex. Still, we use the same tools. I mean, differential equations, ordinary or partial differential equations. We can use also agent-based models, and also we can use AI elements or machine learning elements. The models are for efficacy in the sense that in this case is if we want to correct something that is not working in the body, or we want either disease because of lack of signaling molecule or cancer, then we want to interrupt proliferation. That would be then we need to model all those processes and the organs and the molecular activity and how the organ is responding. And in toxicity, similar toxicity or safety of drugs, drug safety or toxicity are similar concepts. And this can be on target if it is related to the mechanism of action. For instance, if you have cancer drugs and they inhibit proliferation in the tumor, they are also inhibiting proliferation in other tissues. So that could be on target. Or it could be off target when suddenly that molecule that is supposed to interact or to do with a given receptor and to do something very specific is also doing something else that you are not expecting. And this is extremely difficult because you are not expecting this type of toxicity. And that's why the drugs during the preclinical assessment, they are run in big batteries of in vitro and in vivo platforms, because you want to be sure that there is that you know this part, because the on target is expected, we can predict it, we can assess it. The off target is not expected, so this is extremely risky. So you from genetic mutations to so you really need to assess that part. And then, toxicity is quite complex because we have multiple organs, multiple systems. And then what we have is what is called a paradigm shift from scale-specific approaches to multi-scale models. And that's because when we want to model toxicity, we have multiple organs with multiple space-time scales. So we are really in a complex situation. So from molecular scale to the organism, and the time scale also varies extremely. Changes dimension. So it's quite a complex system. And because of that, the most common approach is to work to develop this type of big models, complex models involving multiple organs and systems. What we do is organizing consortiums. And then this just to show the TransQST consortium that just finished this year, and it was just trying to, the target was to build mechanistic quantitative models to support key risk assessment decisions and to understand the safety of drugs in different organs. So the idea is if we know the PBPK of the drugs because of the properties of the drug, and we can understand how that drug is distributed to the different organs, and we can see how we can quantify how the organs are responding to the drugs, the kidney, the cardiac system, the GI, and the liver. Just by understanding how the molecular disruption in these organs caused by the drug, then we can give predictions before we are ready to give predictions before the clinical trials start. And that's very important because the first time you dose a patient is critical because, things can get wrong, and then you really need as much supportive considerations as possible to be sure that the dose you are giving to the patient in the first clinical trial is the right one, and you don't have unexpected behaviors. So this is another consortium, and this just focus on organs. The previous consortium was big, just focus on many different organs. This one, this DILISIM, focus on your liver, just drug-induced liver injury, and just to be able to predict it in advance. And it's supported by the FDA, that is the Food and Drug Administration in the United States. And then we have also RENASIM, that is another consortium, also to understand kidney toxicity, the drug-induced kidney toxicity. This is very interesting because it's a cardiac toxicity. And it's very interesting because this is promoted by the regulatory bodies. It's called a SIPA initiative. And then we know, when I describe the models of Hodgkin's and Huxley, these electrophysiology models, well, these are the basis of the cardiac models. And the idea is that with some in vitro assessments, you can model electrophysiology of the cardiac cells, and even you can understand the electrocardiograms and how the electrocardiograms or the electrical activity of the heart is changing by the drug. So it all comes from these ionic currents at the cellular level. If you understand how those ionic currents at the cellular level in the heart are disturbed, you can understand how the electrical impulses in the heart, across the heart, are going to be disturbed, and then how can you have arrhythmias. And this is something that you don't want in the drugs, so that we can predict it with these models. So this was also part of TransQST and this publication of a group in Oxford working these models. And it's so important and so much work in this type of modeling that the regulatory bodies are now accepting models as proof of safety. No, it's something, and this type of QST, quantitative systems toxicology models, are proof of safety. So you do the in vitro assessments, with these in vitro measurements, you generate the predictions on the electrical activity of the heart, and this is something that the FDA and the EMEA, or the European medicine agencies, they are willing to accept, and they are promoting this type of modeling. So this is very important for us, because it means that modeling is not only in the lab, it's also starting to cross the borders and be accepted by the people approving the drugs. So now I would like to discuss the novel strategies to address drug safety. And these novel strategies are based on, well, we have modeling. If we understand the whole process, how the drug disrupts the body or the organs, we can give predictions. But in many cases, we don't. Because we know very, very little. That's the truth. No, we know practically, well, we know something, but in the whole in the whole thing is little what we know. So it's difficult to develop these models that in biology that can describe all the processes. So you need measurements. And measurements are usually obtained in vitro systems that are very simple, or in animals. But animals are not so great because there are cross-species differences. So what we have now are what is called advanced in vitro cellular systems. And they are practically organs, laboratory organs. I mean, from human stem cells because of the differentiation of reprogramming methods, but you know that we can really develop tissues and organs in vitro in the lab. And this is very useful for toxicity assessment because suddenly you have we call it organs on chips. Suddenly you have a chip that represents or recapitulates all the bone marrow functionality or the gastrointestinal functionality or the liver or the and you can put drugs in the systems and see what is the response of the organ. So they have a large experimental capacity, and then you can have you can collect multiple endpoints. But also the most important part is that they derive from so they are of human origin, and that avoids the cross-species differences of responses because the drugs that we are generating today, they are very specific at the molecular level. And there we have many cross-species differences. Molecularly, the rat and the human are not are very close, but not that close that we don't have differences. So the problem that these systems have is that the output is not always straightforward to understand in a clinical context. I mean, you have a chip, but it's not really the whole organ. You have hepatocytes working more or less in an organized way with each other. And then you measure the number of hepatocytes that are dying. But how do you relate that number of hepatocytes with the liver injury in the patients? Because then in the clinic, what you are going to measure is the production in plasma of a series of enzymes that get elevated when you have some problems in the liver. So how do you connect this number of hepatocytes in my liver, in my chip, my liver chip, with ALT elevation in patients? And there is where modeling has now a massive role, and is really trying to connect to put all those missing parts in the way. No, if you damage hepatocytes, you will have a series of processes that we can model, so that you will have this elevation of enzymes in the patients. So the thing is that the main target to do this type of combination between cell systems or chip organs and modeling is because we want to increase the safety of patients and also reduce animal experimentation. The pharmaceutical industry uses animals, but it's not really a choice. It's something, I mean, it's not by choice. It's because there's no there's no other alternatives. But on the other hand, I think it's the industry doing investing the most to replace these animals because it's really, well, looking for alternatives, not only because of animal replacement, also because the animals lie, so we don't trust them. So we really think that this type of systems, organ on chips, are going to increase patient safety and reduce animal experimentation. So I'm going to show you some examples on how this works. And the first one is, well, in hematotoxicity. So in hematotoxicity, in AstraZeneca, we have this bone marrow on a chip. We have a scaffold that resembles the bone structure. And in that scaffold, we put hematopoietic stem cells, and they proliferate, differentiate, and produce the whole hematopoietic tree, regenerate, I mean, recapitulate the whole bone marrow with all the different cell lineages that then you will find in the blood, like red blood cells, platelets, neutrophils. So monocytes, lymphocytes, all of them are here, no? Because we have the origin, the common progenitor for all of them, so that the common progenitor expands and generates the whole system in this chip. And then, well, there's a microfluidic system in which we have some a medium that is refreshed every 3 days, and it's pumped through the system so that the scaffold is refreshed with a new medium. So, and then we sample and assess that the number of cells and how the different lineages are developing. So we can, it's very easy to understand that once we have the system in the lab, we can have many of the chips working, and we can do very complex experiments by adding and removing different drugs in single drugs or combination of drugs, just to assess how these cells are affected by the drugs. The thing is that this, and this when we say, well, this is great, because what we do is we have this very interesting system, and just let me show you the characteristics of the in vitro system. So you see this, the human bone marrow, and this the scaffold. So it has very, very similar structure, physical structure. And in this kind of caveats is where the cells are going to form the niche, the stem cell niche, and to keep proliferating and differentiating. We know that we have all the cell lineages by single cell RNA-seq analysis. We can see that we have red blood cells, lymphocytes, platelets, neutrophils, monocytes. So we have the whole, the full hematopoietic tree. But only progenitors. We don't have mature cells. We have only the progenitors that are in the bone marrow. Mature cells are in circulating in blood. So this system has only the progenitors. This the bone marrow, what is inside our bones. And also, well, we have that we sample the circulating fluid in the system that you saw in the previous slide. And the problem we have here is that, well, great, we have the bone marrow progenitors here in the system, and we can assess how they move around the scaffold and how they respond to drugs. But then really what we have in the clinical space or setting, what we have is number of neutrophils in the blood, I mean counts of blood cells in patients. So how to connect this, what we see here, responding to the drugs, with the impact in the human, in the patients. So for that, we have mathematical models. And then in this case, we have the mathematical model of the human hematopoietic system. That means that we measure the response of the progenitors in the in vitro system here, and then we implement that toxicity in our system, in our model for the human hematopoietic system that contains all the different progenitors and circulating cells too. So we have not only the progenitors, but also the circulating cells that derive from those progenitors. This is what the mathematical model integrates, and also it integrates the feedbacks from the progenitors from the circulating cells to their progenitors. So it's quite a complete model, and because of this mathematical model that connects progenitors and circulating cells, we can give predictions on the effect of the drug that we measure in this system, how the drug is affecting the progenitors. This part is going to reflect on this part, on the circulating cells, and that's called cytopenia risk. That it means how the blood cell counts are going to go down after the drug is administered. So it's quite a nice setup, and we are really using this very often now. So this, for instance, one of that, just to explain further, how do we do this translation of chip toxicity into cytopenia risk. So we measure in the chip the concentration and the progenitor response. So we know what is the relationship between this concentration and the response of the progenitors, the inhibition of proliferation, if it is oncotherapeutic and cancer treatments. Then we can integrate that relationship in the patient by understanding the clinical exposure, I mean the concentration in plasma in the patient, and this human hematopoietic model. So we know what is happening in this part, so we can then see how it's reflected in circulating cells. And this is what we have here, the predicted circulating cells in the patients regarding, I think these are lymphocytes, neutrophils, reticulocytes, and hemoglobin. So That means that before start clinical trials, if we run or we perform experiments with the drug in our in vitro system, we can give predictions on the response of the patients to this drug regarding blood counts, regarding the impact on the bone marrow. And it's very important for cancer treatments because one of the main toxicities in cancer treatments is hematotoxicities, the decrease of blood counts, platelets, neutrophils, hemoglobin, everything comes down. So we know that that's going to happen, but also it needs to be managed in a way. So you cannot really, if drugs they have to be efficacious. So you have to know that the tumor is affected. Most of the times, in that case, also the bone marrow is going to be affected, but it has to be affected in a way that is compatible with the treatment and with the life of the patient. I mean, you cannot be so toxic that it cannot be dosed. And this, does it work? This type of approach. So if you see here, this carboplatin, that is a cancer treatment, common one, very much used. And this a dose that is common in patients, and then you can see the platelet counts in patients, and the neutrophil counts in patients, and the black lines are the representation of the average of these measurements in patients, that are the blue lines, and our prediction is the pink line. So we think that we are quite close to the black line. That means that for many molecules, we think that before the molecule goes into the patients, we can give a very accurate prediction. So just to show you how we are implementing this type of strategy that combines advanced cellular systems or microphysiological systems, organoids, with modeling in AstraZeneca. So these are the number of drug studies during the years in our team, in the team where I work. So when we started working with this type of microphysiological systems and developing the models, we started with 2 studies in the microphysiological system and 5 studies in the rat. But if you see today, in 2022, last year, we had just 1 study in the rat and 5 studies in the microphysiological system. And this is this reduction in the animal use is good. We don't like to use animals, but it's also good because really the microphysiological system has improved accuracy of our predictions, because it's better in capturing the toxicity, the human toxicity, because it avoids these cross-species differences. So if you see here this number of drugs that we have assessed in the system, the microphysiological system has failed only in 1 of them, for platelets and neutrophils. Where, for instance, the rat, we have failure in platelets practically all the time. So then it's not only that we replace animals, it's also that we increase the quality of the predictions. So that's why we are very excited about this type of approaches. And just to show you very briefly how we are doing the same to assess gastrointestinal toxicity using these advanced cell systems. So oncotherapeutics, they have a bone marrow toxicity. That's a very, very common toxicity for cancer treatment, but also the intestinal epithelium is also very much affected. Also, if you know, the skin is affected, the hair is affected, all those tissues that are actively proliferating are going to be affected by drugs that are inhibiting proliferation. I mean, you inhibit the proliferation of the tumor, but also you inhibit the proliferation of all these tissues that are very active. The hair or the skin can be serious because you can have rashes, and that can be serious. But also the intestinal epithelium is very serious because can cause, after you damage the intestinal epithelium, you get infection, inflammation, and diarrhea. And the diarrhea can be life-threatening when it's very strong, when it's very severe. So these are representation of this intestinal epithelium. So it's this monolayer that covers the mucosa. No, here we have the villi. These are the crypts, and this is the lumen. So in this cartoon, what we have is that this monolayer covering these structures that you see here, well, the stem cells, the epithelial intestinal stem cells are here in the niche that is at the base of the crypt, together with Paneth cells. They proliferate, and while proliferating, their progeny is pushed, because these are very close or very restricted geometry. The new cells are pushed, are forced to move upwards, to migrate towards the villi. So it's a very dynamic system, and this push and this constant proliferation and migration, what it does is renewing, constantly renewing the cells that are covering or are lining our mucosa. And these cells are shed from the tip of the villi. So it's a constant production, migration, and shedding, so that we have a very dynamic, it's this type of system in which you look at the epithelium and it always looks the same, but in fact it's never the same because it's constantly, continuously being renewed. So it's a beautiful system to model for mathematical modeling. That's why the models would really like this this epithelium, because this is very nice to do. It's also a small number of cells with many different types. We have many different types of cells, so we like it. And then the other thing that we have for the epithelium are new technologies to assess it in vitro. Because in vivo is quite difficult. Imagine that in vivo to take biopsies of patients just to assess how the epithelium is responding to the drugs is not possible because that's very invasive. So we are, but now we have new technologies, and among them is these organoids. Organoids are these cartoon of the organoid. They are a little bit like upside down epithelium, in which this could be the crypt. Here we will have the crypt, and this could be the lumen. And this could be the villi. So it's like an inside out epithelium. But let me show you the movie if it wants to start. No, now it doesn't. But let me see. Yeah, now, so organoids, this organoid are growing, and then it starts from a small number of cells that form a cyst, and then from the cyst you get these protrusions here that are the crypt domains, and the stem cells are here in the base of those crypts. So that means that we have a very good system to test drugs, because we just need a kind of petri dish with this with the signaling molecules that enable the proliferation and differentiation and the formation of these organoids. Also, we have 2D form, this the 3D, what we call three-dimensional organoid, but also we have other in vitro systems like the microtissues, in which we just have a monolayer of cells over a stroma of mesenchymal cells, and also it can be tested with drugs. Although, well, depending on the drug, this is more more suitable to understand proliferation because it's extremely proliferative, while this less proliferative is more suitable for absorption assays. So, and this just to show you that in the same way that I was doing for hematotoxicity, well, we are doing the same for the GI toxicity to predict clinical diarrhea associated with oncotherapeutics. So what we do is measuring these organoids in plates. How do they respond to different concentrations of drugs? From there, we understand the relationship between the drug concentration and the proliferation of the organoids. And then we use a mathematical model of the human small intestinal epithelium to understand how this relationship is disrupting proliferative cells in the crypt and how that results deficit of cell migration towards the villus, and then a deficit in the epithelium, and ulceration and inflammation of the epithelium, and diarrhea. So from here, we can predict clinical diarrhea. And this is an example with this 5-fluorouracil or 5-FU, that is a common cancer treatment. And we predict about, at a given dose, we are predicting at about, 30% of diarrhea that would be just after dosing. And the 30, well, we predict 32%. And the measurements indicate, I mean, the clinical studies or the clinical trials, diarrhea is detected in 22% of the cases. So we think it's quite a good prediction. And then modeling well can really guide dose scheduling at the beginning of drug development. And just to show you just our most developed computational model is a model for the intestinal epithelium. And in this case, this is a case that we were discussing at the beginning. If you can know it all, you don't really need even preclinical measurements. You don't need rats or monkeys or advanced cell systems. You just need to understand what's happening. And then you need a good modeler with the skills to put all that moving parts together in an in silico platform. And that's what we have done with Louis, who works with me in AstraZeneca. He's an excellent, very talented modeler, but he has done with the intestinal epithelium. So we know how the intestinal epithelium works. We know the spatio-temporal dynamics in the crypt geometry. So if you see this in silico representation of that proliferation, migration, and shedding of the crypts to the lumen, this would be the column in which the large intestine, in which we don't have villi, we have only crypts. And then we have all the cell lineages, we have the signaling pathways, governing differentiation and proliferation. But more importantly, in this model, what we have done is within each cell, because what we do is modeling each single cell in the epithelium. Within each cell, we have the cell cycle proteins, and we have also the dynamics of DNA and RNA synthesis and metabolism. And that means that when we add the drugs that we know they are going to perturb, to affect, inhibit one of the cell cycle proteins, or they are going to inhibit DNA synthesis, we can do that in each single cell and see how that propagates to the crypt and to the tissue at the top, and into the epithelium in the top of either on the villi or on the surface of the large intestine, and see how that is disturbed and has means that when we have loss of barrier integrity because the epithelium is not there, the cells are not produced, are not there, we are going to have diarrhea, and that can be life-threatening. So here in this part, you can see that we are predicting epithelial injury associated with CDK1 inhibition. CDK1 is a protein of the cell cycle that controls or regulates the mitosis, what is the separation of the cells. So if the cells cannot go into mitosis, what happens is that they become apoptotic because so it's the apoptosis kind of suicidal cell program. It's a death program that the cell takes because it knows it's faulty. So, and these are when we so here will be the crypt, and these the cells in the epithelium, the different cell types in the crypt, and these are the number of cells on the epithelium at the top of the. And what you can see is that we start inhibiting for 4 days that protein, CDK1. So these gray cells that cannot divide, that's why they are big. They start, they are there in the system. And then they move, before they die, they move with but because they are not dividing and many of them are affected, So we have a decrease of number of cells on the crypt, and that means a decrease of number of cells on the V line, and then diarrhea. The most important thing of this is that we are doing this predicting human epithelial injury and diarrhea without data at all. It's just the model and the model knowledge and the understanding of the process. So, and that's, I think, is well, quite important to show that if we really understand everything, the modeling can be extremely useful. And then just this, my final slide, in which, well, it's a comment by James Black, who was a Scottish physician and pharmacologist. There are many kind of statements like that one, but well, I chose this one today, and it's, well, models in biology, what are they? Accurate description of our pathetic thinking. It means that still we don't understand, and that's what is really limiting the developing of good models, is that we don't understand the processes. So it's data, knowledge, research. So we need we need to develop new modeling platforms and technologies, but it's really very important that we get a good understanding of the biology behind the process we want to model. So thank you, and I think we have only 3 minutes left for questions, but I'm very happy to answer any question, if or any comment, or to discuss anything.
Thank you, Carmen, for this interesting talk. So, time for questions.
Hello, this is Gunnar from Linköping. Thank you for the nice presentation. I heard some of it before, actually. So for these models that you talked about in the end, these agent-based models where you have intracellular signaling and metabolism, and you build up and you have this sort of stochastic-looking simulations, was there any type of validation based on those simulations, or was it just output of the model, so to say?
Yes, well, the validation, if you mean comparison with data, yes, we have done that at different, the things that the model has many scales. So, and we don't have data for all the scales. We have data for the number of cells in the crypt and in the villus, and we can see that not in the CDK1 that I represented, because well, that's something that's quite new, but we have done it for DNA damage for the example with 5FU that I presented with the ODE models, but we have the same with the agent-based model, and we can see that at the cell composition level, yes, it agrees, and at the diarrhea risk also agrees. So the problem with the models sometimes is that it's true, we have many assumptions and hypotheses, and we cannot even, there are many scales that we cannot validate because there are no data available, and there will not be data in the near future. But at the scales at which we gather data, we validate and we refine the model if we see that it doesn't agree.
So that brings me a little bit to my second question, which is, so you have all of these many scales and you have all of these details, and you don't have any data for those. So how do you prevent error accumulation? So if you have sort of small errors in many lower models,
yeah, but yes, of course. I mean, it's but that's why what we do is wherever for us, with this we have the model, and it's a beautiful abstract, complex series of concepts that we need to tie down to the earth, no?
Yes.
Down to earth. And what we do is, whenever we have data, whenever we can see that we can get data either in literature or in, it cannot be, and it doesn't have to be quantitative data, also qualitative about behaviors, dynamical behaviors. we try the model to recapitulate, to check that it recapitulates whatever data, qualitative or quantitative, it's there. And that's exactly for that reason. We know that with all these beautiful, complex, large models, we can really go very wrong when things accumulate. But that's why what we do the molecular scale. We try to understand if we are at least representing the right dynamics of the cycle proteins, and for that we have some data. We do the cellular scale. We have some data on how the composition of the crypt and the spatio-temporal dynamics. So we validate it. And then also we have some data about how that reflects into a diarrhea. So we compare predictions with observations as much as possible at the scales at which we can gather data. So we are very, and the thing is that in academia or it's important to compare data observations with predictions. For us, working with patients, it's really important that we don't get it wrong. So we really are focused on just doing models that embrace the complexity, but also models that are accurate.
More questions?
Thank you. This is Giorgio Davico from the University of Bologna. I wanted to ask you one thing, because it was mentioned in a couple of slides that you could use AI and machine learning methods throughout the drug development process. But how do the regulators, such as the European Medicines Agency, see the use of these methods, like in the drug development? Like, do they accept evidences from AI, or what?
I don't know about that, because I don't work. I don't know specifically what is the position of the EMEA or of the FDA with AI methods.
Yeah. No. Okay. So you.
The things that you can use it, and it's used very much because it's the way of understanding and discovering new targets and new molecules and new structures. But then what they require in regulatory bodies, usually what they require proof that you have this molecule and that molecule is safe and is efficacious. They don't really mind how you find the molecule. In a way, once you get the molecule, if you got to discover that molecule with AI and machine learning methods, that shouldn't be a problem at all. I don't know if where we are having problems is when we want to submit, for instance, as proof of safety, just a prediction, like we are doing in the cardiac space. Then is when really the regulatory bodies are not used to that, because they want data, not predictions. But in the case of in the discovery that is very early in development, it's open for you to use all the technologies that you can use.
Yeah. Okay. So I guess it depends on where in the drug development process you would use it. Okay.
Yeah.
Thank you.
If you use it for submission to regulatory agencies, that's another story, and they are not used to that, and that's quite difficult.
Yeah. Okay. Thank you.
It can be that you delay the project.
Yeah.
I just had a question about determining the right dose before entering clinical trials. What are some strategies that are available for determining whether there's a drug-drug interaction that would affect whether the concentration is within that therapeutic index? Are there any potential ways to kind of navigate that and model that?
Yes, that's the main use of these PBPK models, physiologically based pharmacokinetics models. The big model that I show you, well, first, there are a few in vitro assessments that you can do. So you know that if the drug has common clearance pathways, two drugs then can interact with each other. I think that's what you meant about drug interactions. Is that what
Yes.
you are? Yes, no?
Yes, exactly.
So yes, so yes, no, that's very, very important, much more because now it's very frequent, very frequently we use, we combine the drugs, no? And the first and the most important and the first thing to do is to understand the interactions between drugs. So first is you have to understand the metabolism of the drug, and you can do that in advance by understanding the clearance in vitro systems of those drugs. And then you can predict, you see these models that are complex about pharmacokinetics, the physiologically based pharmacokinetics, how the drugs, how one drug is going to affect the clearance or the metabolism of the other, and then the clearance from the body. And we get predictions before clinical trials for that. And predictions are not bad, but the important thing is that they improve constantly. So the models are under constant development and improvement. But that's one of the first things assessed that need to be assessed in drug development. The drug-drug interactions is a very big work in preclinical stage.
Okay, wonderful. Thank you.
Okay, so we should end. So thank you, Carmen, once again for the talk.
Thank you very much to all of you.